Hello friends, welcome back to my channel. Today we will discuss about the particle size analyzer. So the particle size analyzer are essential instruments which is used in laboratories and production facilities to measure the size distribution of particles in a sample. Basically these analyzer helps to ensure product quality, consistency and performance, especially in industries like pharmaceutical, chemical, food and material sciences, right? So let's start. Friends, my YouTube channel name is Pharma Quality. So please view, like, comment and subscribe my channel. So now here first we will discuss the definition of particle size analyzer. So when beam of monochromatic light source is passed through a dispersed sample at an adequate concentration in suitable liquid or gas, the light is scattered by particles at various angles, which is measured by the multi-element detector, right? So basically this describes the core of laser diffraction method, right? So the monochromatic light, that is laser, interact with particles suspended in a liquid or gas. Basically the larger particles scatter light at smaller angles, whereas smaller particles scatter at wider angles, right? So a multi-element detector capture these angular scattering, and then the pattern is analyzed to calculate the particle size distribution using scattering theory, right? So now here we'll discuss why particle size analysis is being done, right? So the particle size analysis is crucial step in various industries, particularly into the pharmaceuticals, chemicals, food and cosmetics, right? And the particle size impacts properties such as solubility, dissolution rate, stability, appearances and flowability. The laser diffraction has emerged as a leading technique for rapid, reproducible, and reliable particle size distribution analysis. So, the controlling particle size ensure consistent product performance and quality. So, in pharmaceutical, it affects the bioavailability and drug release, whereas in food and cosmetics, it impacts the texture, appearance, and stability. So, the particle size analysis is very crucial step to determine these things. So the final product will be the quality one, right? And the laser diffraction is widely adopted because it covers a broad size range from nanometers to millimeters and offers high repeatability and delivers results within a seconds, and making it ideal for both R&D and production environments. So now here we will discuss the different components of the system. So first one is the optical unit. So basically optical unit transmit red laser light and blue laser light through a sample and then use its detector to generate data about the light scattering pattern caused by the particles in the sample, right? And it is the main unit of the instrument and acquire the command given to perform the task. So here you can see this is the optical unit. So this is just image for the elaboration purpose. Now the advanced malvern particle size analyzer is into the market, and this is the very advanced instrument, right? So next one is the dispersion unit. So there are two type of dispersion unit. First one is the wet unit, that is hydro MV, which control the dispersion of a sample suspended within the liquid. So hydro MV will be used for the liquid samples. And the second one is the dry unit, AeroS, right? So AeroS is the model, right? So this is allow a dry sample to be dispensed and evenly fed to the measurement cell within a continuous stream of air. So this dry unit, that is AeroS, will be used for the dry sample, right? So here you can see these are the two models that has been used for the particle size analysis for the different like liquid and AeroS for the dry. Next component is the measurement cell. So the cell is interface between the dispersion unit and the optical unit. The sample is rotated between measurement windows in the cell so that the laser can pass through it in order to make a measurement. So this is the main unit for the particle size analyzer. So here you can see this is the measurement cell and this is the optical unit. And into the new model of master sizer 3000 of Malvern make, so these optical unit and measurement cell combine into the single one. And next one is the master sizes software. So the software is used to control the optical unit and dispersion unit. These all unit will be controlled by this software and also process the raw data gathered by the system. So these are the basics component of this Malvern particle size analyzer, right? So now here we will discuss the sample dispersion methods. So already we discussed into the component of the hardware, right? That is for the dry and wet. So let's discuss about the wet dispersion. So the sample is suspended in a liquid medium, example water or isopropanol. So dispersing agents may be added to the prevent agglomeration, that is suitable for cohesive or agglomerated powder. Right, so wet dispersion ensure particles are well separated in a liquid, minimizing the clumping, and it's useful for the fine or sticky powder that naturally agglomerate, right? So dispersing agents or surfactants help to maintain individual particle in suspension for accurate measurement. Next one is the dry dispersion. So powder is dispersed in air using compressed air or vacuum, which is ideal for the free floating and fragile samples where liquid could alter properties. So dry dispersion avoids liquids together and is essential for the moisture sensitive, fragile or free flowing powders, right? So compressed air or vacuum system break apart powdered clumps and carry them into the measurement zone without altering the sample's properties. So these are the two methods we use for the particle size analyzer. So now here we will discuss the overall operation procedure, that is how to create a method and perform the analysis. So let's discuss about the creation of SOP. So that is method for the hydro MV and AeroS. So here you can see this is the unit for the hydro MV which is used for the wet sample analysis, and this is the AeroS which can be used for the dry powder samples, right? So I am giving you example of the master sizer 3000 software system. So from that you can operate the system and perform create the method and perform the analysis. So for that in the main window you need to select the new icon and then select the SOP option, and there will be the two option like hydro MB for wet sample analysis and AeroS for dry sample analysis. Then as per your requirement you need to select both of the unit, right? Then click on the OK, and now the SOP editor window shall be appear on the screen. So now here we need to enter the details like identification, particle type, material, dispersant, and instructions if you are having any instruction section under the sample tab. Then click on the next section icon and then enter the details one by one in duration, sequence, obscursion, and alarm section under the measurement tab. So these all the parameter will be the part of your standard testing procedure based on the method validation, right? And after that you need to enter the details one by one like necessary and cleaning sections under the sample description tab. Then click on the next section icon and here enter the details one by one for the analysis, results, and user sizes sections under the data processing tab, and then click on the next section icon and then enter the details one by one in data export like data export, averaging, and printing section under the output tab, and then click on save option, right? So once you will click the save option there will be the window appear and you need to assign the file name for to save the method, right? So in this way you can create the method. So now here we will discuss the procedure of sample analysis. So prior to start the analysis, clean the assembly to be used from the accessories functions and the ensure that the background of the dispersion, it should be as exponential curve, that is curve should be smooth, right? So before proceeding for the analysis, you need to clean the assembly. So now here we will discuss the procedure of analysis for wet analysis, right? So from the master sizer software system, from the main window, you need to select the run SOP option, and then select required SOP from the desired path, and then click on OK option, right? And then the new measurement file window shall appear on the screen, and here you need to enter the file name as per the requirement and select the desired path, and then click the save option. So the file will be saved for the wet analysis, and then prepare the sample suspension as per the standard testing procedure, and then fill the tank with dispersant and press start to begin the measurement. So the sample suspension preparation and the dispersion, whatever the dispersion will be used, that should be the as per the standard testing procedure. Now the sample documentation window shall appear on the screen, and enter the sample name, batch number, and click on OK option. And then instrument shall initialize followed by performing alignment and background measurement. So it will automatically initialize and give you the instruction to do. And then the system displays the message like add sample until the ops cursor is in the range. And after when you receive this message, then you need to add the sample suspension till ops cursor is in the range, and click on measure sample option to start the measuring, right? Then after completion of measurement, instrument shall perform clean system as mentioned in the SOP. And for final report, click record view and select the data generation, then click on reports tab to open the report. So in this way you can perform the wet analysis, right? So now here we will discuss the analysis method for the dry analysis, right? So start the compressor unit and allow the compression unit to fill within the air. And prior to start the analysis, clean the assembly to be used from the accessories function, and ensure the background of the dispersion, and it should be exponential curve, that is curve should be smooth, right? Then in the main window select the run SOP, and then select the required SOPs for the dry analysis, and then click on OK option. Then the new measurement file window shall appear, and here you need to put your file name and click on the save option. The sample measurement window appear on the screen, then add the sample to the feed tray and close the lid. Ensure the sample tray contains sample and is loaded into the arrows. Then click on start option. Then the sample documentation window shall appear on the screen, right? And here then enter the sample name, batch number and conditions, and click on OK option. Then system shall align itself automatically and start the measurement. And then finally the final report, click record view and select the data generated, and then click on reports tab to open the report. So you need to follow such step to complete your dry analysis. So friends, this is all about the particle size analyzer basics and operation procedure. I hope you can understand now. Thank you.